I will start in a couple of minutes. Okay, so I'll share my slides. This is the fourth session for this course, NMR Spectroscopy for Structural Biology. In the last class, we had looked at different DNA conformations and how the NMR of DNA would appear. Another very important biological macromolecule is proteins. And in this class, we will be going more into that. So proteins are polymeric compounds where the monomer is an amino acid. And amino acid is a molecule which has an NH2 group, a carboxylic acid, COOH group, attached to a carbon called the alpha carbon. To this carbon, there is another hydrogen and an R group. This R group can be of 20 different types, forming the 20 naturally occurring amino acids. Two amino acids can come together, lose a molecule of water, and form the CONH bond, also called a peptide bond. Like this, many amino acids can come together to form a long peptide chain, which folds into a protein. Now, since this alpha carbon has 4 different substituents, it is chiral, and hence the mirror image is non-superimposable. You can see this is an amino acid and this is the mirror image. They are non-superimposable since this is a chiral carbon. So there are two enantiomers called L and D or S and R. And the L amino acids are the naturally occurring amino acids. Now we will look at the 20 different amino acids that occur in nature. The first ones are the ones with a electrically charged side chain. This charge can be positive or negative. Within the positive, first we have arginine. So arginine, this is the C alpha to which there's an NH2, COH and H. And the R group in arginine is a CH2, CH2, CH2 attached to this group, which is called a guanidinium group. Since it is basic, it can accept an H plus and have a positive charge. Then you have histidine, where the R group is CH2 followed by an imidazole ring. Again, this is basic, so it can accept an H plus to get a positive charge. And the third positively charged amino acid or basic amino acid is lysine, where the R group is CH2, 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 NH2. So there are three basic or positively charged amino acids. And note, for each amino acid, there is a 3-letter code written in brackets and a 1-letter code written like this. There are 2 acidic amino acids, aspartic acid, where the R group is CH2COOH. This H is acidic, so it can be lost and it can become negatively charged. Similarly, glutamic acid is very similar. There is just one extra CH2 group. So CH2CH2COOH, which exists as CO minus, so negatively charged. Then you have the amino acids with polar side chains but uncharged. The first of these is serine, where the side chain is CH2OH. So due to the OH electronegativity, there is a partial charge distribution here, but there is no complete charge like the first group. Then you can have threonine, which is like serine, but there is an extra methyl group at this position. The other 2 polar uncharged amino acids are the ones where there is a amide in the side chain. These are asparagine with a side chain of CH2, CONH2, and glutamine with a side chain of CH2, CH2, CONH2. So these are like aspartic and glutamic acid, but instead of the acid, they have an amide. There are some special amino acids. We'll look at this at the last. Before that, these are the amino acids with the hydrophobic side chains. Hydrophobic generally means they have only like carbon and hydrogen side chains. So alanine, the side chain is only a methyl group. Valine, the side chain is an isopropyl group. Isoleucine, the side chain is a secondary butyl group. Leucine, the side chain is a this kind of uh, group where there is a CH2. This is also a butyl group, but with different branching. Then there is methionine, which is an alkyl chain, but there is a S here. So CH2, CH2, SCH3. Sulfur is not that electronegative to make it fall between the polar uncharged residues. And also due to the 3 CH2 groups, it falls more into the hydrophobic region. Then you have the aromatic amino acids. Histidine is also part of the aromatic amino acids, but it also has the basic character. So it has been grouped under the positive amino acids. Within the aromatic amino acids, the other 3 are hydrophobic, where you have CH2 connected to a phenyl group called phenylalanine. CH similar to phenylalanine, but at the para position, there is an OH, that is tyrosine, and CH2 connected to an indole ring, that is tryptophan. All these form the amino acids with hydrophobic side chains. There are 3 special cases of amino acid. One is cysteine, which contains sulfur. So the side chain is CH2SH. Why cysteine is special is because 2 cysteine molecules can come together, lose their hydrogen, and form a disulfide bond. Then you have glycine, which is the only amino acid which is non-chiral C alpha, because the C alpha has 2 hydrogens. So one hydrogen is already there and the R group is also a hydrogen. So the C alpha here is not chiral. This is the only amino acid with the achiral C alpha. And proline, where this NH2 becomes a secondary amide. So, and forms a 5-membered ring. So this is C alpha attached to N, COH, and instead of the R group being free, the R group folds back and attaches to the NH2 again. So it is a cyclic amino acid where the instead of a primary amine, you have a secondary amine. So when this kind of a peptide bond forms, this NH is lost and it is attached to the next amino acid. So this does not have any hydrogen on the N when it is part of a peptide chain. So these are the 3 cases of the special amino acids. Now the amino acids can, as I said, many amino acids can come together to form a long peptide chain, which can fold into a protein. So proteins have different levels of organization. The first, you can have a simple chain of amino acids. That is the primary structure of a protein, which defines the sequence of the chain of amino acids. This can be folded in different ways. The two most common ways are shown in a helical way, which is called alpha helix, or in a beta pleated sheet. So again, we look at these in more detail here. The alpha helix is basically, you can see this alpha helix, it has been written out here. So this is different amino acids, and in the alpha helix, you have hydrogen bonding. So suppose you start from this N, uh, let's start from this NH. If I call this as I amino acid, NHCO, then again this NH will be I plus 1. Then CO. Again, this NH will be I plus 2. CO, this NH will be I plus 3. I'll just write it, I'll write it here. And this NH is I plus 4. So the NH of the I residue to the carbonyl of the I plus 4th residue, there is a hydrogen bond formed, shown by this dotted line. And so there is I plus 4 to I hydrogen bond in the amino acids, and that happens between every I to I plus 4 pair. And that leads to this kind of a secondary structure called the alpha helix. The beta sheet, basically, there are two peptide chains parallel to each other, and NH of one chain forming hydrogen bonds with CO of another chain. Similarly, NH of the other chain to the CO of this chain. This forms a kind of secondary structure known as beta pleated sheet. So the secondary structure is about the local folding of the polypeptide chain into helices or sheet. These are the most common, but it can be other secondary structures also. So both of these, they have regular hydrogen bonding. Then the next level of organization is a three-dimensional folding of this secondary structures due to side chain interactions. So there's relative orientation of the secondary structures held together by side chain interactions that lead to this tertiary structure. You can see here this red is a beta pleated sheet, this blue is an alpha helix, and they are held together by different kinds of side chain interactions. It can be pi-pi stacking, van der Waals, electrostatic, and so on. The final level of protein structure is the quaternary structure, where proteins consisting of more than one amino acid chain come together. This is said to be multiple domains. So each amino acid chain folds into a different tertiary structure, and each tertiary structure is called a domain. Multiple domains are held together by different non-covalent interactions, and that is the quaternary protein structure. Now we will look more into the different, some other secondary structures. So we looked at helices and beta sheets. You can also have turns. Turns are anywhere there is a change in direction of the chain. Like suppose here it is shown highlighted. So the chain was going this side and then suddenly it turns around and goes this side. This is facilitated by what is called a turn. Turn is this kind of a, you can see again, the chain was going up, this kind of a structure when it appears, now the chain goes down. Similar to the other secondary structures, there are hydrogen bonding between different residues in turns. And depending on the pattern of hydrogen bonding, turns can be of different types. So here you can see the two most common types, type 1 and type 2 are shown. Here, mainly, it is the orientation of, if you see this orientation of the amino acid, this is different in type 1 and type 2. And also CO is pointing inwards in type 1 and it's pointing outwards in type 2. And the NH is opposite. In type 1, it was pointing outwards. Type 2, it is pointing inwards. So these kind of minor orientational changes are what define different kinds of turns. And so how do we define a general turn? Firstly, there will be 4 amino acids which form a turn. So if I consider this as ith amino acid, then from CO, NHRCO, this is I plus 1, again NHRCO, this is I plus 2, and NHR, and continued, this is I plus 3. So the CO of the I residue is hydrogen bonded to the NH of the I plus third residue in this turn. And the distance between these two C alphas can maximum be 7 angstrom for this kind of a structure to exist. Different kinds of turns, like the type 1, type 2, have different relative phi-psi orientations of the I plus second and I plus third residues. That we already discussed here, that it is the orientation here and here that is changing to define the different kinds of turns. Now, within the beta sheet itself, there can be 2 types of beta sheet. First is the parallel beta sheet. So we define direction as N to C terminal. You can see this is the N terminal, this is the C terminal. So the direction of this chain is this side. Again, this is in the same direction, N to C terminal this side. So these 2 lower chains form parallel beta sheet since they are both pointed in the same direction. Then from this chain to the upper chain, it is anti-parallel because this chain is in this direction, but the upper chain, N is here, CO is here. So N to C terminal will be this direction. So these 2 form anti-parallel beta sheet. And again, the top 2, if you see that, this N to C terminal, this is pointing this side, and N to C terminal, this is pointing this side. So these 2 also form anti-parallel beta sheet. Now the hydrogen bonding pattern in parallel and anti-parallel beta sheets is different. In parallel, you have NH to CO and again CO to NH. This forms a 12-membered ring. So if I start counting from this hydrogen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It forms a 12-membered ring. And similarly, for all the hydrogen bonded rings, they are all 12-membered rings, and the hydrogen bonds are slightly tilted. But if we look at the anti-parallel beta sheet, I'll start counting from this hydrogen. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is a 10-membered ring. And the adjacent ring, if I start counting from this oxygen, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. This is a 14-membered ring. So there are alternate 10 and 14-membered rings formed in the anti-parallel beta sheet. And here the hydrogen bonding is much more straight. Because of this, anti-parallel beta sheets are more stable because the hydrogen bond is in the most preferred linear orientation. Now, they can parallel and anti-parallel beta sheets can also be defined like depicted like this, where the sheets are shown by arrows and the sheets are connected by turns like this. So this sheet was going this side, a turn is involved in changing the direction, and now it's going this side. Or if it is parallel, this sheet was going this side. Now a turn is involved which completely goes over the molecule and comes back, and then again there is a sheet. So these is how it can be depicted in proteins and the parallel and anti-parallel beta sheet. Now, when you have a peptide, there are different kinds of torsional or dihedral angles. If you remember, a dihedral angle is the angle between 4 atoms. So if we start from C alpha N, carbonyl carbon C alpha. This is defined as the dihedral angle omega. Now this NC bond, it has partial double bond character because the lone pair of nitrogen is in resonance with the carbonyl double bond. Because of this partial double bond character, this omega dihedral angle can adopt 2 main values, 180 degree, where this is the 180 degree because the 2 C alphas are opposite to each other, and it is called the trans, or 0 degree where the two C alphas are on the same side of each other, and that is called cis. In cis, you can see the groups are, R groups are closer to each other, so there are certain steric clashes. So all the torsions will not be allowed. Certain torsions will be disallowed due to very high van der Waals repulsive effects. This is what I was talking about, the restricted rotation of the double bond. This uh, lone pair on the nitrogen of the amide delocalizes into the carbonyl or double bond. And so this one of the resonance structures has a double bond, which means that the peptide bond has a partial double bond character. And due to that, rotation along the peptide bond is restricted. Now, there are other dihedral angles which we can define. So if we go from N to carbonyl carbon to C alpha to the next N, that is defined as chi. And if we go from carbonyl carbon, C alpha N to the next carbonyl carbon, that is defined as phi. Ramachandran defined a map where chi was plotted in y-axis and phi was plotted in x-axis. And in this map, only certain torsions are allowed. So the regions shown in dark red are the strongly favored torsions. The light red regions are less favored, and all the other regions are completely unfavored because the van der Waals steric repulsions are too high. Within the allowed regions are where you will find the different secondary structures. So if you see this region with phi chi of around minus 60 and minus 40, this is where the alpha helices occur, the left-handed alpha helices. The right-handed alpha helices occur around this region. The beta sheets occur here, and the parallel and anti-parallel beta sheets have different phi chi angles. Turns occur here, and so on. So this is a map which defines what are the torsions allowed for secondary structures. Now going back to NMR, here you have a 1H spectra. The top spectra shows that of a folded protein, whereas the bottom spectra is for an unfolded protein. What happens is that in an unfolded state is a very dynamic structure. It can have multiple conformations for each amino acid. So for each amino acid, you will see an average chemical shift. Hence, in the folded, you can see many more peaks were visible. But in unfolded, due to the averaging, the peaks start merging and the ranges where the peaks are seen become narrower. So that is the difference between folded and unfolded spectra of a protein. Of course, a protein has so many hydrogens from 1H spectra itself, you will not be able to deconvolute anything. You will need two-dimensional or higher dimensional spectra. Now, if we look at a polypeptide chain itself, then different regions of the polypeptide chain will occur in different regions of the NMR spectra. For example, this is a leucine side chain. One second. Sorry, this is, yes. So this side chain that you have, there are methyl groups. The methyls occur below 2 ppm. You can have other aliphatic protons like here, here. These will occur around up to 3 ppm. Then you have the H alpha, which is the hydrogen to which the NH2 and CO is connected. That generally occurs around 3 to 5.5 or 6 ppm. Then you can have side chain NH2. This is a side chain NH2. So they will occur around 6 to 8 ppm. You have the aromatic protons for the residues which have aromatic parts in their side chain around 7 to 8 ppm, and the backbone NH can occur from 7 to 10.5 ppm. These are the backbone NH. So different regions, different functional groups occur in different regions of the 1H NMR spectrum. We will now look at the different amino acids and the different kinds of hydrogens they have. So if we start from proline, This is the backbone nitrogen. This is a tertiary amide, so there is no hydrogen to it. Now you go to C alpha, which has a H alpha, and it is connected to a carbonyl. C beta has two H betas, beta defined as H beta 2, H beta 3. Similarly, C gamma has two H gammas, gamma 2, gamma 3. And C delta has two H deltas, delta 2, delta 3. These are the different kinds of protons in the proline side chain. If you have the glycine side chain, the backbone NH is there, and the C alpha has two different hydrogens, H alpha 2, H alpha 3. Why all these hydrogens, even though they are on the same carbon, they are different, is because uh, of the different chemical environment they exist in, whether they are R or S. Then alanine is there, which has a backbone NH. C alpha has one H alpha and one carbonyl. And C beta is connected to three equivalent H betas. This is the methyl group in the alanine side chain. Then there is arginine, where this is the, again, for arginine, this backbone, this is no, for starting from alanine, this region that is shown in dotted, this is there for all the other amino acids, this backbone NH and the H alpha. So for all the other amino acids, it is shown starting from C beta. So arginine has two H betas, two H gammas, two H deltas. The next is called, after delta, it is epsilon. So the hydrogen on this N is N epsilon. Then you have the next two hydrogens. One is called eta 1 and eta 2. Eta 1 has two different hydrogens, eta 1 1, eta 1 2. Eta 2 has two different hydrogens, eta 2 1, eta 2 2. Asparagine, again, two H betas are there. Then there is a CO and NH2. So it's C beta, C gamma, N delta. So the hydrogens are, if this is N delta, that has two different hydrogens. Then for aspartic acid, C beta has two hydrogens, C gamma, O delta. O delta has one H delta. This H may often not be observed because it is a very polar H and it can exchange with the solvent. Then for cysteine, you have the H betas, and connected to S gamma, there is a H gamma. For glutamine, C beta is connected to 2H beta, C gamma is connected to 2H gamma, C delta, then N epsilon, and N epsilon is connected to two hydrogens, N epsilon 2 1 and epsilon 2 2. For glutamic acid, beta and gamma is similar. Then you, instead of the amide group, you have COOH. So from C gamma, it becomes C delta, O epsilon, so the H epsilon is there. Similar to aspartic acid, this hydrogen may exchange with the solvent and it will not be seen. For histidine, again C beta, H beta is there. Then it comes to the imidazole ring. In the imidazole ring, this is C gamma. Then you have two deltas. This N delta 1 connected to H delta 1, C delta 2 connected to H delta 2. Then from delta 1, you have epsilon 1, delta 2, you have epsilon 2. So the hydrogen connected to C epsilon 1 is H epsilon 1. Hydrogen connected to N epsilon 2 is H epsilon 2. This is the side chain of histidine. And it is important to remember that the side chain of histidine, resonance can be there. So when histidine side chain accepts a hydrogen, there is a positive charge on it. The positive charge can exist either on this nitrogen, or there can be resonance and it can exist on this nitrogen. So there is a distribution of positive charge in the aromatic ring of histidine. Now isoleucine will have, again, the C beta will have H beta, but then it has two C gammas, C gamma 1, C gamma 2. C gamma 2 has three equivalent H gamma 2s, whereas C gamma 1 will have two, C gamma 1 has H gamma 1 2, H gamma 1 3. These are non-equivalent. and it leads to a C delta 1, where again there are 3 equivalent H delta 1s. So whenever there is a methyl group, all the hydrogens are equivalent, but when you have a CH2 group, the hydrogens are non-equivalent. Then there is leucine. Leucine is again C beta connected to 2 different H betas, then connected to a C gamma. C gamma is connected to H gamma, and it is also connected to C delta 1, C delta 2, which are corresponding to 3 H delta 1s and 3 H delta 2s. So again, within the methyl group, the hydrogens are equivalent, but between two methyl groups, they are different. Lysine has a long CH2 side chain. So C beta with 2H beta, C gamma with 2H gamma, C delta with 2H delta, C epsilon with 2H epsilon. And finally, the NH2, the N eta, it has two H etas, or if it has accepted one H plus because it is basic, it will have 3H plus. Next, there is methionine. Again, C beta and C gamma with corresponding hydrogens. S will be delta. So the methyl group after that will be epsilon. So these are the 3 epsilon hydrogens connected to the C epsilon. Now coming to the aromatic phenylalanine. C beta will have 2 H betas, then C gamma. Then there are 2 C deltas connected to H delta 1, H delta 2. Then 2 C epsilons connected to H epsilon 1, H epsilon 2. And finally C eta connected to H eta. Next, there is serine. C beta connected to H betas. So O will be gamma, and the H connected to O is H gamma. Threonine is similar to serine. There is an additional, instead of this H beta, threonine. So C beta is connected to one H beta, but then it is also connected to a C gamma. The O gamma, H gamma remains same as serine, but there is an additional branching here, which is C gamma connected to 3 H gammas. Next, the other aromatic amino acids, tyrosine is very similar to phenylalanine, only instead of H eta, it is connected to further OH. Tryptophan is a bit different. It's like the branching is a bit more complicated. You have C beta connected to 2 H betas and 1 C gamma. C gamma is connected to 2 C deltas. Delta 1 has 1 H delta 1. C delta 1 is connected to N epsilon 1, which is connected to H epsilon 1. C delta 2 has two branchings. C epsilon 2, C epsilon 3. C epsilon 3 is connected to H epsilon 3. Then epsilon 3 is connected to C eta 3, which is connected to H eta 3. C epsilon 2 is connected to C epsilon 2, which is connected, C eta 2, which is connected to H eta 2. And both of them are connected to the next carbon, which is connected to a hydrogen. So there are many different kinds of hydrogens in the tryptophan side chain. And finally, valine, the branching occurs at beta. So C beta itself is branches into, there's one H beta and it branches into C gamma 1 and C gamma 2. And they each have their respective hydrogens. So C gamma 1 has 3 H gamma 1s, C gamma 2 has 3 H gamma 2s. So within the methyl group, the hydrogens are equivalent, but between two methyl groups, they are non-equivalent. So what was the need of studying this? Now, when we look at the NMR of each amino acid, the range at which the different protons appear is different. So if we look at alanine, alanine just has H alpha and H beta. We are only looking at the, we are not looking at NH here. And this OH will anyway go because it is connected to the next peptide. We are looking at the 1 to 5 ppm range. So we are only looking at the H alpha and the side chain hydrogens. Alanine has one H alpha, which occurs around this range, around say 4 to 5 ppm. And the H betas of the CH3, which is occurring less than 2 ppm. So alpha and beta are well separated in alanine. But if you have serine, there is a OH connected to the CH2. So OH is an electron withdrawing group. It will withdraw the hydrogens towards itself. So the beta CH2 here will get more downfield shifted in serine compared to alanine. So if we look at serine, alpha is around in the same range of alanine, around 4 to 5. But beta in alanine, which was below 2 ppm, in serine, it is greatly downfield shifted to around 3.5 to 4 ppm. So alpha and beta are not well separated in serine at all. There is a much greater downfield shift of beta hydrogens in serine compared to alanine, where they are much more upfield shifted. This is because of the electron withdrawing of the OH group of serine, which makes this beta hydrogens more downfield shifted. Compared to serine, if you look at threonine, similar effect will be there because again this OH group is there which is withdrawing electrons. So again alpha and beta are not well separated in threonine, but there are additional gamma protons in threonine due to this methyl group. This is the gamma methyl group. So methyl groups are quite upfield shifted generally. So the gamma protons occurred around less than like around 1 ppm here. So in threonine compared to serine, threonine has an extra set of protons that are the gamma protons. So like this, each amino acid has its characteristic, its side chain protons occurring in characteristic regions of the 1H NMR spectrum. Just 1 second. Okay. Now, so as I was saying, the protons of each amino acid side chain occurred in distinct regions which are characteristic to that amino acid. Now, toxispectra is used for spin identification. We have already seen this. So in toxi, it's a two-dimensional spectra of 1H versus 1H, and you will see all the connectivities. So you will see H alpha to H beta to H gamma to H delta, and so on. So um, if you look at each vertical line, it represents one amino acid. So in toxi, we'll start from here. This is the leucine. So this is the H alpha H beta correlation. Then you have H beta with H gamma. Then you have H gamma with, toxi means that you will see from, let me just go up here. So if it's leucine, you will see alpha to beta and alpha to gamma and alpha to delta. So toxi will show all kinds of correlations from one hydrogen to the rest of the hydrogens in the side chain. So if I'm starting from one particular, let's say I'm starting from H alpha. So H alpha to H beta, H alpha to H gamma, and leucine has two different H deltas. So H alpha to the two different H deltas. Here also there are two because there are two different H betas. So H alpha to the two different H betas that is shown here. You can see there are two different H betas in leucine. Similarly, for other amino acids, if you just take the along a vertical line, it will show all the correlations within one residue. So each amino acid will have a characteristic pattern of cross peaks. Sometimes the H epsilon or H delta, which are like H epsilon, which is very much away from the H alpha, or H delta, which is also quite away from H alpha. Sometimes these may not be seen if the mixing time is short, that is, the magnetization is not allowed to travel that far from H alpha. So sometimes the protons which are more down the side chain away from H alpha may not be observed in toxi. Cozy is similar to toxi, but it only shows adjacent spins. So cozy will show H alpha to beta, beta to gamma, gamma to delta, whereas toxi will show alpha to beta, alpha to gamma, and alpha to delta. Toxi shows much longer range correlations. So that is why in cozy, suppose we are looking at this region 7 to 9, where you will see the NC alpha correlations. So each amino acid will have one NC alpha cross peaks. There are 5 cross peaks, so there are 5 amino acids. Whereas toxi, you will see many other additional peaks because this NH, so NH is basically here before the C alpha. Let me write it like this. So NH will show with H alpha, H beta, H gamma, H delta. Due to all these additional cross peaks, toxi has much greater peaks. You can similarly, this was in the 7 to 9 ppm. Similarly, you can have in the aliphatic ppm regions to see the correlations in the aliphatic regions. Toxi will always have much more greater peaks than cozy, since there are much longer correlations visible in toxi. Now we will look at the cozy or toxi of each amino acid separately. First, we look at histidine. And here we are ignoring the NH because these spectra are recorded in D2O. So if there is an NH or OH or COH, that proton will exchange with the deuterium. And we know deuterium is NMR inactive, so it will not show any peak. So all these NH, OH, COH, we are ignoring here. So this is the histidine side chain. And again, here we are looking only in the side chain region of the, you can, so we are only looking at the correlations within the side chain. So here you will see these are two hydrogens in the histidine labeled 2 and 4. They are not equivalent because 4 has a nitrogen on either side of it, but 2 has only one nitrogen on one side of it. So 2 and 4, they will show correlations in toxi. So this 2 hydrogen is written here 2H, 4 hydrogen is written here 4H. We know the diagonal is representative of 1D spectra. So if we can write the 1D, the two different hydrogens on the diagonals, and they form a cross peak due to this correlation. Now you can have tyrosine. In tyrosine, you have so this is called the AX system because you have two different kinds of hydrogens. In tyrosine, A prime, these are equivalent. These are both. Let us first start from. So you have A prime, which are ortho to the OH group, and X prime, which are meta to the OH group. So this system is called A prime, X prime. They are, why are we not calling both as A? Because they may be equivalent, their chemical shifts are the same, but their J values are not equivalent. So it is better to call them as A prime, X prime. But their chemical shifts are same. So A prime will form just one peak, which is written as here, and x prime will just form one peak, which is shown here. They will have a cross peak due to this ax or a prime x prime correlation, which can be considered as cozy or toxy since they are adjacent, which is this cross peak. Then the phenylalanine ring. Ortho to this position, you have x prime. Meta to this position, you have a prime. And para to this position, you have m. So here the system is a prime, x prime, m, which is written here. Again, we are not making both the ortho or both the meta as equivalent, because although their chemical shifts may be equivalent, they may have different J values. But since chemical shifts are equivalent, they will occur in the same region. So the two ortho have one peak. Para has one peak. 2 meta has one peak. So here the ortho, so we can say, so these are the cross peaks that we'll see basically. One second. So if we'll just label the positions. If this is 1, this is 2, this is 3, this is 4, this is 5, this is 6. So 2 and 6, that is x and x prime, are here. 4, that is m, is here. And 3 and 5, that is a and a prime, are here. So a prime will show a cross peak with m. A with m, a prime with m. Then a prime and x prime will show a cross peak. A x, a prime x. That is this cross peak. This cross peak, which is between the x and x prime to m, it will not be seen in cozy because it is not adjacent. Cozy will show only adjacent. But it can be seen, it will be seen in toxy. That is this x to m and x prime to m, they will be seen in toxy. So it is shown like this kind of cross. That is, it will not be seen in cozy, but it will be seen in toxy. Now, if we look at the tryptophan side chain. Tryptophan, this NH is exchangeable. So this hydrogen does not have any other hydrogen it can couple with because this is exchangeable. So this 2H, it appears as a single dot. There is nothing it can couple with. Then you have these adjacent protons, they will couple with each other. So say 5 and 6, 4 and 5, and so on. So basically there are 3 couplings possible in cozy. This, and this. These are shown by the white dots. In toxy, there may be some other couplings, like toxy can show between this and this, between this and this, between this and this. So toxy will show 3 additional couplings between what we can call the metas and the paras. So there are 2 metas, this and this, and this and this, and the paras. Toxy can show all possible couplings, which are shown by this cross. These are the 3 toxy couplings, 1, 2, and 3, which are seen in toxy but not in cozy because they are not adjacent. So this is about the aromatic amino acid spin system identification. Next, you can have so serine, cysteine, aspartic acid, asparagine, phenylalanine, histidine, tryptophan. All these, now we are looking at the aliphatic regions of these. These all form, these 8 shown here, all have a similar pattern in the aliphatic region. That is AMX, which is alpha and 2 betas. So all of these have H alpha and then two H betas. OH we'll ignore. Again here two H betas, COH we'll ignore because it's exchangeable. Asparagine also, H alpha, two H betas, NH2 also is exchangeable. Similarly, cysteine, SH is exchangeable. Tryptophan, it has H alpha, two H betas. Rest all form in the aromatic region, which we have already discussed. Now, right now, we are only looking at the aliphatic region. And same for the other aromatic amino acids. So these are AMX system, where A is alpha and M and X are the two betas. So alpha will show correlation with one beta, alpha will show correlation with the other beta, and beta will show correlation between each other. Here toxi and cosi are same because alpha and beta are adjacent. So these 8 are AMX systems. Here are some with some different kind of branching. So valine has alpha, beta, and then two sets of gammas. So alpha, beta, then gamma CH3, gamma CH3. It can be written as A3B3MX system. A3B3 refer to the two gamma CH3s. MX refers to alpha beta. So in cosi, you can have alpha to beta, beta to one gamma, beta to the other gamma. In toxi, you can have alpha to gamma also and alpha to the other gamma also. Then for leucine, there is alpha to two betas, and then there is one gamma hydrogen and two delta CH3s. So this is written as A3B3, which refers to the two delta CH3s. MPTX refers to the four different protons, the gamma proton, the two beta protons, and the alpha proton. Here the cosi will be alpha to one beta, alpha to the other beta. Then beta between the two betas. Then from one beta to the gamma, from the other beta to the gamma. And from gamma to one delta CH3, gamma to the other delta CH3. These are all the cosis. Toxi will refer to all the other like long range correlations. So alpha to the gamma, alpha to the two H deltas, beta to the two H deltas, the other beta to the two H deltas. So these are all the toxic correlations as well. As for isoleucine, there is H alpha, one H beta, one CH3 gamma, two other H gammas, and one CH3 delta. So this system is called A3MPT, then B3, then X. These A3, B3 refer to the gamma CH3 and delta CH3, and MPTX refer to the two gamma hydrogens, beta hydrogens, and alpha hydrogen. So this is how isoleucine is written. And here also, if we look at the correlations, for cosi, you have alpha to beta, beta to one gamma, beta to the other gamma. Then you have between the two gammas. You also have beta to the gamma CH3. Then from gamma, you have to the delta CH3, and from the other gamma, you have to the delta CH3. There is no correlation between this CH gamma and the other gammas because they are more than 3 bonds apart. So if I start 1, 2, 3, then to the gamma will be 4 bond. So that is not observed in cosi. And all the other in between correlations, the long range correlations, are observed in toxi. We will look at the remaining amino acids, which are called the long side chain or type U spin systems. These include lysine, arginine, methionine, glutamine, glutamic acid, and proline. So glutamine, glutamic acid, and methionine, they are similar in the sense they have two H betas, two H gammas, and one H alpha. We will not consider NH2 because it is exchangeable. Again, glutamic acid has 1H alpha, 2H beta, 2H gamma. We will not consider OH because it's exchangeable. And methionine also has 1H alpha, 2H betas, 2H gammas. We don't consider this CH3 because the magnetization is not able to be transferred there. These are called AM bracket PT X systems. So you have alpha, 2 gammas and 2 betas. Here, betas are more upfield shifted than gammas. This is why these three are different, because normally you have uh, alpha, then beta, then gamma. That is but here what you have is that betas are more upfield shifted than gammas because gamma is attached to electron withdrawing group. So gamma is attached to CONH2 electron withdrawing group, or COH or SCH3 electron withdrawing group, and gamma gets more downfield shifted towards alpha. So that you can remember that this is the difference in pattern. So after alpha, instead of beta, gamma comes. Correlations are as expected. Alpha will show with the 2 betas in COSY. Gamma of one will show with the 2 betas. Gamma of the other will show with the 2 betas. And toxi will show the long range ones also. So alpha with the 2 gammas. For proline and arginine, the delta hydrogens are more downfield shifted than beta and gamma due to the same reason. So in proline and arginine, if we look at H delta, it is close to the electron withdrawing nitrogen in proline, or for arginine, the H delta is close to the electron withdrawing guanidinium group. So the H delta is closest to alpha. Normally, beta is closest to alpha. In glutamine, glutamic acid and methionine, gamma is closest to alpha because gamma is attached to electron withdrawing group. In proline and arginine, delta is closest to alpha since delta is attached to an electron withdrawing group. So here also you have 1H alpha, 2H beta, 2H gamma, 2H delta. So here one is shown because often as you go down the chain, the differentiation reduces and the 2H deltas appear very close to each other. So for COSY peaks, alpha will show with the 2 betas, each beta will show with the gamma, gamma will show with the delta. Toxi will show all other cross peaks, like alpha will show with delta, alpha will show with gamma also. Beta was already showing with gamma, it will now show with delta also, and so on. And you have lysine. Lysine, the epsilon is attached to the electron withdrawing NH2. So it is the epsilon which is closest to the alpha. And lysine also, you have alpha. For COSY, alpha is correlating with the 2 betas, beta is correlating with the 2 betas are each correlating with the gamma, gamma is correlating with delta, betas are correlating within each other, and delta is correlating with epsilon. And if you look at toxi, all the long range correlations are also possible. These 6 form the long side chain or type U spin systems. Just 1 second before I go into nosy. So we were talking so far about toxi and cozy. Now let us look at nosy. Nosy, if you remember, are correlations between nuclei that occur close together in space within 5 angstrom. They are not connected by bonds, but the coupling happens through space through dipolar coupling. There are three kinds of nosies. One is the near neighbor interaction, that is I to I plus 1 or I minus 1, that is sequential. So D alpha n means alpha of I to n of I minus 1. So here, if you look at it, let us look alpha of I, sorry, D alpha n is between alpha, sorry, this is, 1 second, this will be I plus 1. So D alpha n is from alpha of I to n of I plus 1. Similarly, alpha of I plus 1 to n of I plus 2. These are called near neighbor since it is to the next sequential amino acid. We don't consider within the same amino acid because this is also a cozy cross peak. Then DNN is here, n of I to n of I plus 1. Or if you have n of, if there was an I, let me start with I plus 1 then, n plus, n of nh of I plus 1 to nh of I plus 2, or nh of I plus 1 to nh of I. Again, only sequential, the one immediately before or immediately after, it's DNN. Or D beta N. Beta N is not similar to alpha N, but for the betas. So it is not shown in this picture here. But you can imagine it like if there is a 1 second. So this is C alpha, this is beta. So if you have NH to the, say this NHI plus 1 to the C beta I, that will be D beta N. You can see it is shown by the arrow, it's not labeled, but this is D beta N. Again, it's sequential. You can have short range interactions, which are 3 to 4 residue apart, shown by the dotted arrows. These determine the secondary structures. For example, D alpha N, I plus 2. So alpha of I to N of I plus 2. DNN, I plus 2. So N of I to N of I plus 2. D alpha N, I plus 3. So alpha I, N I plus 3. D alpha N, I plus 4. Alpha of I, N of I plus 4. So all these dotted arrows, they are short range interactions between residues that are 3 to 4 residues apart, and these determine the secondary structure. We'll look at long range interactions a little bit later, and these are the ones that are 10 to 15 residues apart, and these occur due to folding of the protein. Different secondary structures have different values of these distances. So you can see the secondary structure is shown here. Each have characteristic values of these distances. And we know nosy intensity is directly proportional to 1 by r to the power 6. So depending on the distance, the nosy cross-peak intensity will be different. And you can see that if you remember what we learned in nucleic acids, that following the nosy spectra, we can determine the what is the sequence of the nucleic acid. We can do the same for proteins as well. So in a helix, this basically each this DNN, that is between say NH of I to NHI plus 1, this DNN in helix occurs for each I. There will be two cross peaks with I minus 1 and I plus 1, which will be observed, and there will be one self peak with the uh, uh, no, NN will not have any self peak. NN will only have, so the different amide hydrogens are shown uh, in this diagonal. Each amide hydrogen NH will have nosy cross peaks with the NHI plus 1 and NHI minus 1. So if we start from the terminal, it will only have one cross peak, because suppose I'm starting from one end. So it will, if it is I, then it will only have I plus 1. There is nothing before it. So there is one cross peak. Then this is the self peak of I plus 1. I plus 1 will have two cross peaks with I and I plus 2. And like this, if you follow the nosy cross peaks, you will be able to determine the amino acid sequence. Similarly, for other secondary structures as well, if you follow from cross peak to self peak and so on, walk along the sequence, you will be able to determine that the primary structure of the amino acids. Here we are looking more closely into alpha helix. So alpha helix, it will suppose this is a 7 residue alpha helix. So you have 1 to 7 residues. Now, if it is about DNNI plus 2, so 1 will show with 3, 2 will show with 4, 3 will show with 5, 4 will show with 6, 5 will show with 7. 7 is the last residue. So there will be 5 DNNI plus 2, and indeed 5 are shown here, 1, 2, 3, 4, 5, and between which residues they are occurring is shown. So if it is from 1 to 3, the line is from 1 to 3, and so on. If you, then there can be D alpha N or D alpha beta between I plus 3. If it is I plus 3, so 1 will be with 4, 2 will be with 5, 3 will be with 6, 4 will be with 7. There are 4 possible I plus 3, and indeed 4 are shown here for the D alpha N I plus 3, 4 are shown here for the D alpha beta I plus 3. Then there will be the alpha N I plus 4. If it is I plus 4, 1 with 5, 2 with 6, and 3 with 7. So there will only be 3 I plus 4, which are shown here. These are all short range interactions, 3 to 4 residues apart. And the D alpha N or DNN, these are immediate neighbors. That is I with I plus 1 or I minus 1, and these help to determine the sequence. This short range interactions help to prove the secondary structure, and this kind of a pattern indicates that it is alpha helix. Similarly, for other secondary structures, they have very characteristic patterns. This is the long range interactions that we were talking about, which is like 10 to 15 residues apart. So suppose you have a polypeptide chain and you have nosy between this A residue and D residue. That means the chain is folding like this, where A and D are coming close to each other. Or if there is a cross peak between A and B, then the chain is folding like this, where A and B are close to each other, so the nosy can be observed. Or if the chain is folding that B and C are showing a cross peak, then it is folding like this, where B and C are close to each other. So depending on the long range interactions, we can determine the folding of the protein. So from sequential connectivities and short range interaction, we can determine the secondary structure. From sequential connectivities, we can determine the primary sequence, primary structure. From short range interactions, we can determine the secondary structure. And from long range interactions, we can determine the folding of the protein, that is the tertiary structure. Now for structure calculation, we have already seen in the previous class what is the basic concept that is used. So just 1 second. So from the NOE intensity is directly proportional to 1 by r to the power 6, where r is the distance between two protons. So from NOE distance, we will get a range within which our distance should exist, an upper bound and a lower bound. Now the energy is defined by the summation of two terms. One is the intrinsic energy, which depends on the bond distance, bond angles, and torsional angles. And one is the ENOE, which is defined as the sum of two terms. One term is the deviation of our distance from the lower bound, squared and multiplied with the force constant and summed up over all the uh, distances, and another is the deviation of our distance from the upper bound, squared, multiplied with the force constant and summed up over all the distances. If all the distance constraints are fulfilled, then the ENOE becomes 0. That is, all the violations are removed. So in any optimization algorithm, the optimization needs to keep on happening till the ENOE becomes 0. We had also discussed the Tandy algorithm, which is torsion angle based distance geometry algorithm. Here we start from many different initial secondary structures because we don't want the calculation to be biased by the choice of the initial structure. Then each initial structure is optimized by putting the distance and torsion constraints that we obtained from NMR. The optimization is continued till all the constraints are satisfied and we reach the final optimum minimum energy structure. We can see that although we started with different uh, different initial structures, we end up at similar final structures, which indicates that this optimization has been accurately done. Now, we have discussed this before also, that when the spectra gets too crowded, then the dimensionality is increased. For large proteins, the noisy spectra gets too crowded, that for proteins more than 150 residues. Here, another dimension is added, which is the 13C. So basically, you can see here nosy, this is the, so firstly, if nosy is too crowded and we want to use 2D itself, we use HSQC, the 15N 1H HSQC, not 13C. In the 15N 1H HSQC, there is only one peak per residue. So that clears up the spectra a lot. But this is not a better solution is increasing the dimensionality of the experiment. We have discussed this before also. Suppose we have a 1H toxi spectra and a 1H15N HSQC spectra. So here 3 peaks refers to 3 amino acids and they are occurring on the same plane. If we want to separate out each amino acid in different planes, we add an additional dimensionality. So 1H is there. The third axis we are taking as 15N. Here each plane has one amino acid and within the plane, the alpha, beta, gamma of that amino acid is appearing as separate peaks. Even this, if you want to separate out, another axis is added, the 13C for the fourth dimension, and alpha, beta, gamma, they separate out into different planes. So from a very crowded spectra, you get a well-separated, well-resolved spectra, and that is the advantage of multidimensional NMR. We have discussed many different triple resonance experiments. I won't go into the details again. The basic thing is that the name in triple resonance experiment, these are for the three-dimensional NMR, and basically there is a transfer of magnetization between different nuclei and you detect the nuclei and this is different for different experiments. Depending on the name of the experiments, you can understand where the magnetization goes. So HNCA means it goes from amide hydrogen to amide nitrogen to the C alpha. This C alpha can be of the same residue or the previous residue. HN, if there is something in bracket, it means that is involved to transfer the magnetization, but it is not directly detected. So HNCOCA Means again amide hydrogen nitrogen to C alpha, but using CO as a method of transfer. Since in this direction there is no CO, here the transfer can only happen to the previous C alpha. Similarly, HNCO, HNCaCO, these are all in the main chain. You can also have transferred within the side chain where it can go to the C alphas or C betas, and those can also be detected. So these are different triple resonance experiment, and the main thing is 3 different nuclei are detected. That can be 1H, 13C, 15N, or 1H and 2 15Ns, and so on. And these form the basis of the 3D NMR. One experiment, HNCA, we will, so HNCA and HNCOCA, we are just looking into those. We have already discussed this before. We are just brushing it up a bit because these are important mostly in case of peptide NMR or protein NMR. So HNCA, you start from H of I residue and go to the N of I residue. From N of I, you go to C alpha I and C alpha I minus 1. That is shown here. And then you go back from C alpha I and I minus 1, you go back to Ni and again back to the H of the Ni. So these are the 3 time axes that we are taking, or frequency axis: Ni, N, C, and H. So here, if we take, basically, if we take the, say, H versus 13C, we have seen before that if we have a 3-dimensional spectra, F1, F2, F3, we can take different cross sections along these spectra. Suppose we are taking this vertical cross section, which is at a particular value of F1. So F2 and F3 are changing. F2 and F3 are the 1H and 13C. And F1 is fixed, that is at a particular value of 15N. So here we will get one self peak with itself and one sequential peak with the previous residue. We have seen here also that I and I minus 1 appear together. So if we are taking Ni, we will get self peak with I, sequential peak with I minus 1. If we are taking I minus 1, we will get self peak I minus 1, sequential peak I minus 2. So it is always with the previous residue. Like this, by taking different cross sections along the plane where F1 is constant, we will get one self peak, one sequential peak, and we can lay out the different cross sections and go from self peak to sequential peak to again self peak to sequential peak and so on, in order to find out the polypeptide sequence. HNCOCA, so here maybe you might not be sure which is the self peak, which is the sequential peak. In that case, HNCOCA can be used, where you will only get the sequential peak. The C alpha is detected only for I minus 1. So that is helping you distinguish between the self peak and the sequential peak. We have already discussed that experimental 3D NMR appears like this. And if you project the 3D NMR along one plane, it will give the corresponding 2D spectrum. For example, projecting along this plane is giving you 1H versus 13C. Projecting along this plane is giving you 1H versus 15N, and so on. So projection of 3D spectra on the corresponding plane will give that 2-dimensional spectrum. Now, with development of more and more refined NMR methods, the number of protein structures that are being deposited is increasing rapidly. You can see this graph where this orange is the number of structures released annually, and the blue is the total number of structures that are now available. So over the years, more and more and more structures have become available, since more and more refined NMR methods are available to find out structure of different proteins. Now, there are some proteins that pose challenges. One is the intrinsically disordered protein, also called IDP. Here what happens is that the HNCA experiment becomes very difficult to resolve because C alpha is not very well observed in the spectra. You can see for uh, uh, on unfold, suppose this is the folded HIV protease. If it is unfolded, you can see the peaks are not very well defined. It is difficult to understand them. In that case, HNN is used because you can see the nitrogen dispersion. So if we look at the y-axis, nitrogen dispersion, it is retained, but both the 1H, it was 1H was spread out over a longer range, it is now in a shorter range, and 13C get reduced a lot. So instead of using 13C, an extra nitrogen axis itself is used, that is the HNN experiment. Here what happens? We start with the hydrogen of the I residue. It goes to the nitrogen of the I residue. So hydrogen of I to nitrogen of I. From nitrogen of I to C alpha I and C alpha I minus 1. Nitrogen of I to C alpha I, nitrogen of I to C alpha I minus 1. Up to here, it is the same as the HNC. Now it varies. From C alpha I, it goes to Ni plus 1. So C alpha I, it goes back to Ni, it goes forward to Ni plus 1. And C alpha I minus 1, it goes to Ni and Ni minus 1. C alpha I minus 1, it goes back to Ni and it goes to Ni minus 1. And then from the three corresponding ends, it goes to three hydrogens. So Ni, it goes to Hi, Ni minus 1, it goes to Hi minus 1, Ni plus 1, it goes to Hi plus 1. What we are detecting are this hydrogen axis, this nitrogen axis, and another nitrogen axis here. So this is the HNN experiment, and it is mainly used in cases where the 13C dispersion is not very good. So instead of using 13C as an axis, an additional nitrogen axis is used. One of the cases where it is used is the intrinsically disordered proteins. Now, again, if we look at the HNN experiment, the spectra will appear. So this is a three-dimensional spectra. We can take different cross sections of this. Suppose we take a horizontal cross section that is at a particular value of F2, that is F1 and F3 are varying. Then we will see one self peak, that is I with I, which is shown in blue, and two sequential peaks, that is I with I minus 1, I with I plus 1, shown in red. If we take a vertical cross section at particular value of F1, similarly, there will be three peaks like this, one self peak, I with I, and two sequential peaks, I with I minus 1 and I with I plus 1. So this experiment is often called a triplet filter through HSQC because three consecutive amino acids are getting filtered out of the entire polypeptide, the I minus 1 and I plus 1 residues. So, but in HNN, the problem is that how do we distinguish that? So between I and I minus 1, I plus 1, we can distinguish because they have different signs. So if I is plus, I minus 1, I plus 1 will be minus. But how do we distinguish between I plus 1 and I minus 1? There the HNCN experiment comes. Here what happens? We start from the hydrogen of I, it goes to the nitrogen of I. Then it goes to the CO of I minus 1. It is going to CO of I minus 1. From CO of I minus 1, it goes to C alpha I minus 1. C alpha I minus 1. From C alpha I minus 1, it goes to Ni and Ni minus 1. So Ni and Ni minus 1. And then it goes to the corresponding hydrogens. So Ni to Hi, Ni minus 1 to Hi minus 1. This is if we start with I. Now if we start with I plus 1. So hydrogen of I is ending up with hydrogen of amide hydrogen I is ending up with amide hydrogen I and I minus 1. So amide hydrogen I plus 1 will end up with amide hydrogen I plus 1, amide hydrogen I. So this will give us an idea of the directionality, because here only the peak with the previous residue is seen. So if it is I, you will get self peak with I and sequential peak with I minus 1. If you're taking the F1, F3 plane. Or if you are taking, looking at the I plus first residue, then you will get self peak I plus 1 and sequential peak I. So this H and Cn will give an idea of the directionality of the polypeptide, because here only the previous residue is seen, whereas in H and N, both I minus 1 and I plus 1 are seen. Now there are certain patterns in HNN and HNCN that helps determine the uh, polypeptide sequence. So first we are looking at HNN. The general case is YXZ, where YXZ are 3 amino acids which are not glycine or proline. So now the self peak is shown as a square, and the black color means positive. So X with X, it is a self peak and it's positive. And X with Y and X with Z are sequential peaks shown as a circle, and they are negative shown in red. This is the general case. In the H and Cn, you will only see if you take the F1, F3 plane. So if you take the F2, F3 plane, you will see I with I minus 1. But if you take the F1, F3 plane, you will see I with I plus 1. So that is interesting. Depending on the plane you take, you will see with the previous residue or the next residue. So F1, F3 plane will show I with I plus 1. Here we are looking at F1, F3 plane. So X will show self peak, and it will show sequential peak only with I plus 1, that is Z. Then if we look at YGZ, here you have glycine in the middle position. Now, whenever glycine is there, the pattern of signs change. Normally, the self peak is positive, but in glycine, you can see the self peak is red, that is, it is negative. Not only that, the sequential peaks are normally both negative. But in glycine, the sequential peak with I minus 1 is negative, but with I plus 1 is positive. So in glycine, the pattern changes. And another thing is that glycine is generally very much occurring towards the top of the spectra. So the distribution of peak is also different. Now, YGZ for H and Cn, uh, again, it will only show self peak, and it will show with Z, which is I plus 1. Again, the pattern is different. Instead of one positive, one negative, both are positive. If glycine occurs in the I minus first position, like GXZ, in that case, you have X with X, that is a self peak, X with G and X with Z, these are the sequential peaks. Here again, glycine is occurring at the top of the spectra, and instead of both sequential peaks being negative, the one with glycine is positive. So the sign pattern is changed. Here again, if X will show with X, the self peak, and Z will, X will show with Z, that is the I plus 1 peak. The sign, so although it is not showing with glycine, because glycine is the I minus first residue, and we will see only with I plus first. But because of the presence of glycine, the pattern of the peaks, the sign has changed of the peaks. So although the cross sequential peak is not seen with glycine, glycine can act to change the sign of the pattern. You can have two glycines also. You can now, if you have proline, proline does not have a NH, and so there will be no backbone NH, so there will be no cross peak with proline. So X and X, there is a self peak. X with Z, there is a sequential peak. But X will not show any peak with proline because proline does not have any H with which to combine. But so one peak is less when there is a proline in the I minus first uh, uh, position in HNN. But if the proline is in the I minus first position, there will be no difference in the H and Cn because proline is I minus first and that is not involved. So wherever proline is, now if you have two prolines, so PXP, so it will just have self peak, no sequential peaks because proline don't have any backbone NH. Similarly, PXP for H and CN. If you have G surrounded by two P's, again you will have only one peak G with itself, but normally self peak is positive, the glycine self peak is negative. So similarly, whenever glycine occurs in a sequence, the sign of the sequence will the sign pattern of the sequence will change, and that will help you understand that this is a checkpoint in the polypeptide. So suppose you are assigning a polypeptide sequentially. If suddenly the pattern changes, that will prove that there is a glycine here. So here a sequential walk along a polypeptide is shown. We are looking at H and C and spectra. So we will have one positive self peak, one negative sequential peak. Again, we go to self peak, then from positive self peak to sequential peak, from sequential peak to self peak with the next residue, and so on. Now, when we encountered a glycine, let us look at this sequence. It is IGQ. So glycine is in the middle position. It is like YGZ. So then you will have two positive peaks. You can see two positive peaks. Or if we have, say, GQD. So glycine is in the IMAS first position. Then you will have two negative peaks. Indeed, you have two negative peaks. So wherever glycine is occurring, the sign pattern will change. Instead of 1 plus minus, you are having 2 plus or 2 minus and so on. Just 1 second. Right. So this H and CN, it assigns very fast in just a few hours. And we have seen that in HNCN, if we look, so far we were looking at the F1, F3 plane. Because over there, you will be able to see self peak, I with I, and sequential peak, I with I plus 1. If you look at I minus 1, you will see self peak, I minus 1 with I minus 1, and sequential peak, I minus 1 with I. Now, if we look at the F2, F3 plane, which is shown here, these kind of act as glycine checks. That is, in the F2, F3 plane, we will see only the peaks for the glycine residues and the residue next to them. So, suppose we are seeing, let's say, glycine 10, we will see uh, glutamic acid 11. Then glycine 47, we are seeing lysine 48. Glycine 75, glycine 76. So all the glycines will be observed. Some other amino acids will also be observed, which are adjacent to the glycine. So this is a very quick way to check if we have the correct protein, because in a protein, if you have an expected number of glycine and you can see those particular peaks for glycine, and glycine peaks are very uh, like distinct for their chemical shift. So you will know that yes, you have the correct number of glycine. So the F2, F3 plane is used for glycine checks, and the F1, F3 plane in this HNCN we have already discussed. It is used for sequential connectivities. So there is a positive and a negative. So there is a positive for the self peak and a negative for the sequential peak in every cross section that you have taken. When you have glycine, the pattern changes. This C is basically an expansion of this region, which is shown here. Suppose you have PGK. Glycine is in the middle position. That will be this one, YGZ. In that case, both the peaks will be positive. Indeed, you can see both the peaks are black, it is positive. Then if you take the next three, GKT, glycine in the I- first position. That is like GXZ, both peaks are negative. Indeed, both peaks this is connecting to is negative. Or if you have LTG, glycine in the I plus first position, that will be that option is not given here, but the sign is supposed to change. So it isn't that clear here. But for the other two, as I discussed, the sign clearly changes. So TGK, instead of one positive, one negative, you have both positive, both black. And GKT, again, you have both negative, both red. So wherever glycine is occurring, the sign is changing. And the F1, F3 plane is used to determine the sequential connectivities. Now we have discussed about the basics of proteins, peptides, and their NMR. We will look into a few practice problems. The first problem is asking, as always, I'll discuss the problem, give some time, and then discuss the solution. First problem is asking, what is the chemical shift range of backbone amide protons? So here, if you remember this that we had discussed, the range at which different protons in the peptides occur. The backbone amide protons occur around, say, 7 to around 11 ppm. The closest range is A, 6.5 to 10.5, that is option A. It is important to remember that it is different from the side chain NH, which occurs slightly more upfield around 6 to 8 ppm. So that is different. And within this same region also, kind of overlapping from around 6.5 to 8.5, say, you will get the aromatic protons. So you can remember the range and you will be able to answer this kind of questions. 1 second. And for the other ranges, H alpha is a very characteristic range around 3 to 5.5. The aliphatic region around 0 to 3 are where the CH2, these kind of CH2, CH protons occur. And methyl groups have very characteristic range below 2 ppm. If you remember in DNA also, ATGC, the thymine had an extra CH3 group which was occurring below 2 ppm, and that was very characteristic of thymine. So peaks below 2 ppm are very characteristic of methyl groups. The next question is asking, which among the following residues does not have side chain proton resonances in the range 6 ppm to 7.5 ppm? So for questions like this, it is important to remember the different amino acid structures. So if we look at the options given, we have asparagine, so this is the H alpha. The structure is the side chain of asparagine is CH2CONH2. For lysine, the side chain is 4 CH2s followed by a NH2. For valine, it is an isopropyl group. And for glutamine, it is similar to asparagine with the extra CH2 group. So CH2, CH2, and then CONH2. And it is important to remember the range in which different protons occur. Now, 6 to 7.5, we have to remember that it is saying side chain proton resonances. Valine only has aliphatic protons, so it will not occur in this range. So the answer is valine because that is what the question is asking. All others have backbone NH, asparagine and glutamine have backbone NH, CONH2, this backbone. So that will occur around 6 to 7.5 range. Lysine also has a backbone NH, so it will also occur along this range. Valine is the only one which has aliphatic uh, uh, protons, so it will occur much more upfield. So the side chain proton resonances of valine will not occur in 6 to 7.5. And it is important to see the question. It says does not have. So the correct answer is valine, that is C. The next question is asking which of the following dihedral angles of a peptide can be obtained from 1H NMR spectra. So here we'll look at the different dihedral angles first. If you remember from one C alpha to the other C alpha, so let me just, this dihedral angle is omega. So C alpha, C and C alpha, this is omega. Then if you from one n to the n, C alpha, CON, this is chi, and CON, C alpha, CO, this is phi. Now, there is an equation called the Karplus equation, which relates the 3J coupling constants with the phi dihedral angles. It is defined as J, the coupling constant, is A cos square phi plus B cos phi plus C. If you plot this J of two hydrogens with phi, it will give this kind of a curve with a minima around 80-82 degrees, where the J is minimum. So from NMR, we can get the 3J coupling values. And using the 3J coupling values, we can calculate this phi dihedral angle. So the correct answer is A, that is phi. This is not a concept I discussed in the class, but I'm discussing now. So if you remember in the structure calculation, we were talking about distance constraints and torsion constraints. Now the NOE intensity is directly proportional to 1 by R to the power 6, where R is the interproton distance. So NOE intensity will give you the interproton distance constraints. Karplus equation will give you one of the torsion angle constraints. So from NMR J values, you can calculate the phi torsion, and it will be one of the torsion angle constraints. So from 1H NMR, directly you can get the phi dihedral angle. So the correct answer is option A. The next question is asking that which of the following statements is correct? So here, if you look at the different statements, one is that NOE is useful only for peptides around 1 kilodalton molecular weight. That is completely wrong, because NOE is very useful for proteins up to say 150, 200 amino acids. Even after that, it is useful. Maybe it gets a bit crowded, um, and 3D spectra might have to be used for some residues, but there is no such weight limit for NOE. So this is incorrect. C is saying Toxi aids in spin system identification. That is correct, but gives correlation with neighboring residues. This is not correct. Toxi can do, suppose we start from NH, it can go to H alpha, H beta, H gamma, H delta. Toxi will give the entire correlation. So just not between neighboring residues. If this was cozy, this sentence would have been correct. HSQC gives the fingerprint of proteins or peptides. This is correct because HSQC is about 15N1H correlations, and each residue will have one peak in the spectra. So it is kind of like the fingerprint of the protein or peptide, because suppose we have 10 peptides, each with different amino acid sequences, then you will get 10 different HSQC spectra, where each residue will correspond to one peak in the HSQC. So HSQC is the fingerprint of peptides and proteins. That is the correct answer, which is B. The next question is asking which of the following experiment is more useful for assignment of an intrinsically disordered protein? Am I the asshole? So here the correct answer is HNN. If you remember, we were talking about the intrinsically disordered protein, which posed some challenges. Basically, the proton dispersion is very much reduced in these proteins on unfolding. You can see proton was from 7 to 9. Here it's hardly somewhere between 8 and 9, a very short range. The 13C dispersion also gets lost. However, the nitrogen dispersion, 15N dispersion is retained. So instead of taking HNCA, where there is H and C, both of where the dispersion is lost, it is better to take HNN, where the 15N dispersion which is retained is taken in two axes. So the H and N is the most useful for such problem spectra which are of the intrinsically disordered protein. So the correct answer is B. The next question is asking which nucleus among the following is the starting point for magnetization transfer in a three-dimensional HNCA NMR experiment. So here the answer is 1H. One thing to remember is that almost in all experiments, except a few sidechain detection experiments, the starting point is always the 1H. From here, the magnetization travels to the 15N of the same residue and then to the C alpha of the same residue and C alpha of the previous residue. After which the steps are retraced, that is from C alpha of both these, it goes back to N and then to H. So the starting point and ending point are both 1H. And the question here is about the starting point. So the correct answer is B, that is 1H. The next question is asking the 3D HNCO experiment correlates the chemical shifts of which nuclei? So here, if we look at the HNCO experiment, I've already discussed that from the name of the experiment itself, you can answer these questions. So HNCO, it starts from the amide hydrogen, goes to the amide nitrogen and to the carbonyl carbon. So the H and N are of the same residue if we call it I. The carbonyl carbon is of the preceding residue, that is I minus 1. So if we look at the options, Ni plus 1, this is wrong. This is all three same residue, this is wrong. This is saying Hi, Ni, COI minus 1. This is correct. So the correct answer is option C. The next question is asking how many backbone amide cross peaks will you see in the 1H 15N spectra of this peptide in aqueous solution? So here, if you write out the peptide, you will see it is asking about the backbone amides. So if you have if you so for basically in 1H15 and HSQC, you will see one peak for each amino acid. So how many amino acids are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You are supposed to see 12. But what you have to remember here, they are only talking about the backbone amides. So E to G will be 1, then G to F 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. There are only 11 amide bonds, and so there will be 11 backbone amides, uh, uh, which we are supposed to see. That is what it appears to us. Now let us write out the spectra. So we are starting from, let's say, here, which is glutamic acid. So this is 1, 2. Now the thing to remember here is proline. Proline will not have the backbone NH. So you're supposed to have 12. But since proline does not have backbone NH and there is one proline in the sequence, you have 11. I just counted 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. We counted 11. So 11 we were supposed to have, and we have proline. So proline does not have backbone in it. So you will actually only have 10. That is option B. So option B is the correct answer. We'll leave this now. And I will, as always, I'll upload the recording and you can go through it and ask me your doubts. And next class will be the final class of this session.